Okay, thank you very much, everybody, and I would like to really congratulate Francesca and her team, all the sisters and Raffaella, to putting up a very nice meeting. It's a really great pleasure to be here and talk to everybody. We don't get, I personally don't get very much chance to talk to parents and patients. I mostly spend my time in a laboratory. So today, I'm just going to talk to you about a hypothesis. We call it a hypothesis because we still need a little bit more proof. This work hasn't been yet peer-reviewed, but over time we're gaining perhaps a little bit more confidence that this might be real. But please take it as it was not yet scientifically reviewed, has not been published. So I'll just start that there is a considerable overlap between genetic causes of intellectual disability and epilepsy, and if you just look on the number of known genes, there are probably about 600 plus genes causing intellectual disability and more than 400 genes causing epilepsy, and some of these genes cause both, and that's what progesterone 19 fits. So it's really quite complex, and we can say that in many cases the intellectual disability and epilepsy have common cause. We discovered the PCDH19 gene through studying these large families, and I have to say we have been quite lucky to find that gene mutations because the gene was not well characterized and it was not in the right place where it meant to be, or the human genome was not well defined. And then Eric and his team and Christelle Depienne identified mutations in a different kind of patients, which was triggered by the finding of that one single male, which I believe, but please correct me, is the only male which is known with PCDH19 disorder for now. And that's because it pretty much mimics the situation in a female because it's a somatic mosaic. And then, of course, they found mutations in these situations where the mutation is inherited from a father. It can be de novo, and as you heard, we're finding also these situations where the mutation is inherited from non-expressing or non-penetrant mother or grandmother. And that's really very interesting because we can take advantage of these situations perhaps for understanding what's happening in this disorder, what's protecting some people or what's making some people vulnerable to the mutation. This is just a quick summary of the clinical data, but I don't want to go through it because you will have a whole session about it. But really what was quite interesting in this disorder was the age of onset and offset, that these things do happen at some stage, and they actually stop happening in terms of at least seizures at the age above sort of 12 years, around puberty. And that was very interesting. But it was also interesting that the treatment really didn't work in general, but again, you're going to hear about that a little bit more, so I don't want to make any conclusions. And perhaps the most interesting from the scientific perspective, because this has perhaps broad implications for other disorders, was the fact of why only the heterozygotes, or the mosaics in case of males, would be affected. Why actually people can live normal life without having this gene, despite the fact that this gene is actually very well conserved, so other organisms have it, and there is a good reason perhaps why in evolution this gene remained very similar across different species. So if we conceptualize it, I mean, we see really on the tip, and that's where we started, we identify the mutation, and after that what happens, it's really not that obvious to us, because there is a cascade of the primary and secondary events which then lead to this complex biological systematic organismal presentation of the disease, which can have different phases and different facets. We see that the seizure and the severity of the phenotype vary. Sometimes there is early onset, sometimes this disease never onsets. And of course, you end up either affected or unaffected. And there is a spectrum. So it's really quite complex, multi-dimensional space we're trying to understand. And starting from this little tip, which is the PCDH19 mutation, and trying to discover what happens after that, which gives us the outcome. So really the question is that what is the most important? It is the mutation. It is what happens next behind the cloud, or what we see at the level of the patient, because that also depends where we pay attention or how we try to fix it or address it. And that certainly has not been trivial, and not just in this disease, but across many other disorders. So that's why we started with a battery of different tests. We have been working on this probably over 4 or 5 years. So we started with an opportunistic approaches. So in a way, we looked at the cells and just done a few experiments just to see why these cells and how these cells differ from the cells of normal people. And I'm going to talk about that. We do still study families, and that's very valuable. We took advantage of the mouse model, where this gene is not functioning, and I'm not going to talk about that. That has been challenging. The mouse doesn't behave the same way as the patient, so that's the bottom line. So in a way, I'm just repeating what Ricardo said, that the mouse is not always the best model, but in certain circumstances, in certain types of experiments, is extremely useful, and we just ought to use it. We started also on the iPS cells. As Ricardo mentioned, it's a huge expectation and importance to have this resource, because that's the human relevant cell type which we want to study. We look at the yeast to hybrid interaction, so in a way we try to implicate or guess about the role of this protein by looking at which proteins it mingles with, interacts with. I mean, it's in a human society the same way. You can tell about a person by the number of and type of friends they have, what they're likely doing. So that's exactly what we're trying to do here. And we also took another approach, which is another way to tell us what's going on. It's a metabolomics, to look not at genes, but to look at the metabolites in these cells. So I'm mostly going to talk really about the expression profiling for this talk. So we used skin fibroblasts, and that was not because necessarily you can generate iPS cells from them, but also because PCDH19 was expressed in this cell type. So we thought that was relevant to look at all genes which were expressed in this cell type and try to see which genes were different and significantly statistically different, actually expressed in patients versus control. And that's what we were pretty much doing. So we've done it in different stages and for different reasons. We used not the latest technology, but the older technology of exon arrays. Now, these days we would do RNA sequencing, which is more sensitive. But again, the arrays had some advantages. I don't want to go to technicalities. And we used about 12 girls with the mutations. We used controls. We used the males as well as control males. And just to cut it to the chase, we found about 94 genes which were different according to the certain criteria. Initially, it really didn't hit our eye, but when we looked more carefully, we found that almost half of them, or 43 out of 94, were genes which normally show different amounts in men and women. We all differ because of the steroid hormones and the sex-determining SRY gene, which kicks in early and determines whether we're boys or girls. Well, not that gene, but the presence of it or not. And that triggers a cascade of different events. I mean, the expression is also different because of the X chromosome content. As you heard, some genes on the X chromosome escape X inactivation, and the women would have more of those products than men. And of course, there are genes on the Y chromosome also. They are less relevant. However, there was one protocoadherin, which is brain expressed and in human specific. So we had to pay some attention to it, but pretty much the work of Eric and Christelle, by finding the mosaic male, kind of excluded that gene from the picture of being likely the compensatory, the savior of the males. So it looked like going to be something different. So we found this surprising uneven distribution of genes which are normally expressed differentially in different sexes. And when we statistically tested it, it was highly significant because there is not many of these genes. But there is a, they call it cottage industry in science. It's a small group of investigators who are really interested in this because for many years science ignored actually the differences between sexes. In many experiments only males or only females are used, but mostly males. And they differ. we are different. Just look around the audience, we are different. But when you look at the brain and the structure of the brain, the function of the cells, the expression, this is different. So we found that likely that was significant. Now, what this meant. So we started to look at individual genes, and on our lists were genes like glutamate receptor, which in the affected girls, these are these two bars, was much lower than in normal controls. Actually, in males, this gene is very lowly expressed in skin fibroblasts, almost not detectable. And this gene has been significantly associated with epilepsy, not directly by mutation, but has been deregulated in different types of epilepsy. Now, oxytocin receptor, I mean, that's more sort of, if you mention oxytocin, it's more rings a bell about pregnancy and delivery. But oxytocin receptor mice is actually one of the reasonable models for autism, or it has been until probably Ricardo and others generated other mice. And the oxytocin receptor, again, shows very interesting expression. I mean, it's really in the affected girls, it's more highly expressed, and then in the control girls, and the boys have more of it. And then there was another set of genes, these aldo-keto reductases, aka C1, C3 genes, which again were lowly expressed in the affected girls, with PCDH19 mutation, quite highly expressed in controls, and relatively low in males. But from this, if you look at it, the pattern was, and that was striking, that's at least caught our eye, and that's in our bias, that the girls with the PCDH19 mutations looked more like boys than control girls. And that's pretty much the very simplified bottom line of these studies. And that really brought us to pay more attention to these type of genes, these aldo-keto reductases, which are involved in neurosteroid metabolism. Now, neurosteroids are steroids, just they are produced also in the brain or primary in the brain. For many years, it was thought that steroids, these type of steroids are produced elsewhere, but not necessarily in the brain. But it has been shown that in hippocampus and cortex, there is brain-specific production of these cells, and they can be produced in neurons as well as other cell types in the brain. Now, the original fold changes were quite dramatic. These were corrected by real-time PCR and other experiments, but they were significant, and they seem to hold across different series of patients. But what is also important to be sure that this is meaningful result, because we, at the end, study trying to make assumption about brain by studying skin. And interestingly, skin is one of those tissues where these steroids actually can be synthesized. So the apparatus, the metabolic pathways are present in the skin. So that gave us a little bit more confidence that it might be on the right track. Now, very recently, actually, I discovered it relatively recently, but it was about 2 years ago, it was a group discovered that these genes are actually involved in so-called backdoor pathway to dihydroxytestosterone. So actually, you don't need to synthesize testosterone, this dihydroxytestosterone, which is the active neurosteroid, via testosterone. You actually can go via progesterone to get to it. And they discovered it by actually finding mutations in these genes, in case of, in this case, C2 and C4, in these boys with problems of sexual development. So in a way, these boys have X and Y chromosome, but they develop as girls because they have mutations in these genes. Now, what is interesting about this, and I don't want to go really to this problem, I don't want to imply that these girls are in fact boys, because really the expression is likely different only in specific cell types, and we still need to convince ourselves that actually in the human brain and neuronal cells this expression is different, because we don't have the data yet. This is mostly in skin fibroblasts, so we're eagerly waiting for the iPS-derived neurons so we can test some of these hypotheses. But this convinced us that really that there is a significant downstream effect of neurosteroid signaling as a consequence of compromised function of these genes. Because that's what our hypothesis is based on, that if you have small amount of this gene and enzyme activity, then the downstream signaling and it kind of acts as an amplifier effect because many genes are regulated by steroid hormones. So this is just a pedigree just to show that some of these some of these boys pretty much grew up as girls. Interestingly, not all of them were affected because not all of them had all mutations. And interestingly, the girls with the mutations developed as girls, so it affected only boys. Now, we looked at the function of these enzymes, not only at mRNA level, but we also look at the level of the protein. And here you see the Western blot of different affected girls, and you can see that while the level is variable, some may have almost a normal amount with respect to the controls, but there are a few which have very little or almost absent, you would say. So there are changes. But that's, again, there is some normal variation in expression of these genes. And from my point of view, I can say maybe that's why some of them are more affected and the others are not. But of course, there can be many explanations, and one of them can be the X activation, which we heard from Alexandra. Well, then we also tested the sort of the activity of these enzymes in skin fibroblasts. Because there is not much activity in skin cells, we had to really use very sensitive technique of mass spec measurement, and we had to stimulate these cells with progesterone, which is the metabolite, this enzyme process. And after that, we got these results which show quite a bit of variation. We probably need to do more of them. But certainly the cells from the girls with the PCDH19 mutations were very poor metabolizers of progesterone into allopregnanolone. And then with the help of Francesca and Nicola and others here in Rome, they did a very preliminary measurement of allopregnanolone in the blood of these girls, and this is the result. I mean, take it as really preliminary, which needs a little bit more validation. But there was a suggestion, perhaps, that what we see in the level of the cell lines, it might be real. It's really allopregnanolone levels vary, depends on which developmental stage. They can be individually variable. It's a quite complex system which we need to take into account. But on the other hand, it's a system which is quite a good candidate to explain the variable expression of the clinical presentations in these girls. So the signaling, it's really complex, but generally it starts from progesterone or testosterone. But the bottom line is really, this is probably one of the best known and the most potent modulators of GABA receptors. And they've been studying for a long time. Then this is not new, that these neurosteroids, especially allopregnanolone, also tetrahydroxycorticosterone, have been really targeted by many people to be as potential treatment strategies for different types of disorders, not only epilepsy, but also anxiety and stress and depression. Now, there is also evidence that this likely works into at least some models. So I just picked a few. Frank Hoy, our colleague and collaborator, treated this particular type of epilepsy in this Fragile X syndrome, which is another known syndrome, in a mouse. So just single administration of ganaxolone, which is a synthetic variant of the allopregnanolone, just pretty much led to the abolishment of the seizures. Another group used allopregnanolone in neurons, in this case hippocampal neurons, where again the allopregnanolone in this case just returned the cell into a normal status. And most recently, this has been only reported not a long time ago. It was a result of a small symposium, which I didn't attend, but I heard about that work, especially Mike Rogowski. They treated this novel onset of refractory status epilepticus in a 3-year-old or 26-year-old male. His seizures were just refractory to any treatment, but by application of allopregnanolone, they pretty much stopped the seizures. And apparently, as Mike Rogowski says, the man is still seizure-free. So there are a few bits of evidence. Now, the other, I just go back, I showed this last time. These are all the dots of different girls with the onset of seizures, and you see it starts about 10 months. But that's kind of soon after the mini-puberty, when during pregnancy, the allopregnanolone levels go really through the roof. And that's simple because allopregnanolone confers neuroprotection. And also allopregnanolone levels sometimes go up with the stress. So it's a kind of a response of the nervous system. These neurosteroids just help the cells to cope better with the stress. And the onset is about 10 months, and the offset, where it is known, because I think we have many more girls which are still too young, so we don't know when they will stop having seizures, so we don't know, but it's about 12 years. Again, it's about the time when the hormones go up. When you look at another example, and that's the epilepsy associated with menstrual cycle, the catamenial epilepsy, again, you see that the most vulnerable stages during that is in the menstrual, sort of at the edges of the cycle. And women are generally protected during the luteal phase. That's again when the allopregnanolone levels are fairly high. So that's generally quite often used. And of course, it would be interesting to ask the question, so how these girls with PCDH19, when they get to have their cycle, whether they get better or worse with respect to which stage of the cycle they are. And again, that can be individually different, but it can also maybe provide some clues once it's studied properly. Now, the GABA, well, the allopregnanolone acts through the synaptic GABA as well as the extrasynaptic GABA. It's generally synthesized from cholesterol in different types of cells, and have probably, they speculate that it's more important through the extrasynaptic action on GABA. And because these GABA receptors actually stay in the neuron after the seizure, while these GABA receptors are mostly internalized after the seizure. So that's why, I guess, originally the ganaxolone and some of these treatments was targeted to these GABAergic receptors to try to control the seizures. And the other important thing is that they have two different types of action. They can actually go directly to the nucleus, like with progesterone receptor, and regulate gene expression. So it can have quite a multiplier effect because many genes are regulated this way, or can directly bind to the GABA GABA receptor subunits and confer seizure protection. And as Maria mentioned, the fact that PCDH19 interact with GABA is perhaps coincidental. I really don't have an explanation why this would fit together, but I just mention it. Now, other perhaps supporting evidence, which I'm not going to go to detail, is that when you knock down progesterone receptor, you also deregulate PCDH19. This has been published. We just look into the data. And that would link PCDH19 to progesterone receptor. What is also interesting, when you knock down sort of progesterone receptor, you deregulate, you kind of protect the susceptibility to seizures that way, which again implicates the same pathway in a seizure generation. Neurosteroids are also very important in stress situations. So, but it's well known that neurosteroid synthesis, it happens de novo in a specific places, and it's in response to stress. The other disease which perhaps suggests that these neurosteroids are important is Niemann-Pick disease. It's lysosomal storage disease, where actually the cholesterol metabolism is affected. And when they really tried to rescue the phenotype in the mice, they actually succeeded to ameliorate the phenotype to a large extent using allopregnanolone. Again, that was not tested, more than I know, in human subjects. And of course, the interaction with GABA, and I can tell you, but I'm not going to show you, it's really preliminary. We may have a direct link between PCDH19 and progesterone receptor using the yeast to hybrid system. We found another interacting proteins which perhaps link it together, but that's, yeah, early days. So going to the mechanism, because that's the most intriguing, I mean, we need to understand that, at least to some extent, if we want to understand the disorder and tackle it and try to help these girls. So again, I picked the same picture, which is modified from Christelle Depien's and Eric's work, where you have these situations where everything is fine when you have the gene, but everything is fine when you don't have the gene, which we assume that if there are girls which have both copies lost, they're likely going to be okay, but we are yet to find that type of individual. And then there is the situation where you have this mosaic population or cell which then generated cellular interference. This mechanism has been studied quite well in another disorder of craniofrontonasal dysplasia, which is another X-linked disorder, which is ephrin-B1 is involved, and it's again it's a cell-cell communication molecule, which is important. But in this case, I guess there is a question whether the situation is like this. So in a way, the mutant cells form one network and the normal cells form another network, and these two networks send conflicting messages to the center, because one say run, the other one say no, stay, relax, and one of them wins probably every so often. Or there is a situation like this where actually these cells communicate, no problem, and there is a consequence later on. I personally think if this was really the case, you would say that maybe the MRIs and some of the imaging techniques would have picked it up, that there are structural problems. But that doesn't seem to be the case, but maybe we need to look more carefully. But it's hard to say. It can be mixed of both, so I don't want to say it's one or the other. But I kind of like that, that actually these two cells do communicate, but the problem happens on the level of the communication. When one cells have the protocoderone, the other one doesn't. And as a consequence, these two cells respond to signals differently in terms of their gene expression. And because if it feeds into the neurosteroid system, it's kind of like an amplifier. It's just like you put oil into the fire. You just get the sudden explosion of action you don't want to have. So they do contact each other, we believe. As a consequence of those contacts, some cell will have a different expression than the others. And maybe that leads to different consequences, perhaps in migration or their identity. So they end up in a wrong place in the brain, as you saw it also from Maria's presentation, or they become different type of neurons and they form different connections. And then you have more of one type of action versus other, and that leads in regular intervals to the seizure activity. Now, that the migration is likely, the consequence has been actually determined already by these people a couple of years ago who studied PCDH19 in zebrafish. And they also found that it interacts with encadherin, which is another big molecule in the field of neuroscience. But what they found is that when they looked at the cells on the left-hand side are the cells of normal fish, and here are the cells of the fish where the protocoderone-19 was removed using genetic technologies. And here they stain these nuclei, so you see only balls. That's why you don't see the whole cells. These cells behave differently. So these are every 2 minutes. These cells are captured, and over time it's made a movie from it, and that tells you how the cells move in the head of the fish, and tells you also where they end up with. But to make it a little bit more obvious, they also put these trackers, and here you see the normal situation, and here you see the situation in the fish without the PCDH19. So really the cells just kind of like don't know where to go. They lose a little bit of their orientation and perhaps also identity. And when you look really what it means, what it happens to the zebrafish brain, when you look at it, it does look different. Again, this is not necessarily the situation in human patients, but well, we heard that it's not always the same in other organisms than in humans. But it gives us perhaps a clue where we should look for the differential answers. So that's probably would be the explanation. So I was trying to tell you that this is very biased. It's a hypothesis. It's a one explanation. That when you look at these skin cells of these girls, we found that there are many genes deregulated. We have also other candidates. There is aldo-keto involved. There are genes, quite interesting genes in regulation of gene expression, which can also act as multipliers. So this is only one facet of the phenotype. This may not provide the answers. But we thought that we can't really study everything. We had to focus, and we focus on this system because of the capacity and the knowledge about the allopregnanolone neurosteroids in these type of disorders likely to be involved. So that's our bias, and we just have to live with the criticism we're going to get. Why not to study something else? The ACAR1C genes are also the genes which are not necessarily conserved. So in mice, they are different. So maybe that would be an explanation why the mouse is different to human patients, because these genes certainly work slightly different in different organisms. And there is a different number of these genes too. So we checked the the protein, mRNA protein levels and the enzyme activity via the progesterone stimulation. We didn't check the enzyme activity itself. We haven't done that. That's probably still need to be done. And there is some evidence that at least the synthetic allopregnanolone analog, ganaxolone, has been used in human subjects, and including that one person, which the author said is the first human subject which was treated for epilepsy with allopregnanolone only earlier this year. So when we look at all this scheme, really, that we're trying to go the way from here to really pick up all these pieces and decide which one we're going to tackle, whether it's going to be, I don't know, the seizures or the autism or other behavioral problems, it's very difficult. And that's really the case. In this case, if that's the pictorial way to demonstrate how we think, is that perhaps we are just addressing, using this hypothesis and this approach, only a small volume of the complexity in the outcome or the consequences of these mutations. And that's perhaps how we should look at it, that these genes and this particular pathway, if it's going to play a role, it's still going to play a partial role. It's not going to be the complete picture. And that's really difficult to figure out, what everything is going to be important. And of course, we're looking at through the development and whether we can put the water back into the jar after it's been spilled. That's a very good question. I'm hopeful that we can put at least quite a bit of water back into the jar and make these girls do better. But whether we can completely reverse it, that's really the question everybody would like to know the answer. We don't know. So, yeah, what we try to fix. Gene therapy, yeah, there were lots of approaches with here. I don't think it's the way to fix. And is it going to work? If in a 10-year-old girl we're going to try to fix the protein 19, might be too late. So we probably need to fix the secondary and primary, secondary cascades and deal with the outcomes. And I guess that's a challenge. So we need to keep that in mind also when we understand these disorders. And that's why we need multiple models. We need human cells, we need animals, we need to really look at the patients very carefully. And that's going to be the afternoon session. You're going to hear about that. So it's really tough. I mean, I wish we all had, from the podium, the people who present and look after these patients have better news. But I guess we need to be honest also to ourselves that really we're dealing very complex issues. And I think in this project in a short time has been very challenging, but perhaps there is a promise. I have to say, I worked on 40 other different genes, but maybe this is the first one when I really see a promise of potential help. So that's the hope which is driving us. So these are the people and probably many others who helped at different stages and contributed to this work. So I would like to thank them and also for your attention. Thank you.
Thank you, Professor Gecz. Are there any questions for this interesting? Ci sono domande per questa presentazione? Go ahead.
Hello, you mentioned that 12 was the offset of seizures. I'd read and heard it was way older at the end of education, literally 16, 17. And also, was there a percentage that carried on having seizures, or did you find in your research that seizures stopped?
No, as far as I can comment on it, but again, you probably hear more about that this afternoon from the people who have a lot more knowledge about collecting these patients. Some patients do continue to have seizures until later in their life, but I would say this might be a smaller proportion, and we certainly in the familial cases which we studied, there were very few individuals who would have few seizures later in the life, like in 20s. But I don't know if anybody from the clinical colleagues who do this would like to comment. Generally, they stop sort of at around puberty or slow down significantly.
We have to say that unfortunately the proportion of patients who stop having seizures completely reaching puberty is very small. The majority of patients unfortunately keep on going seizures. What might really become less frequent is the cluster of seizures and the severity of seizures and the occurrence of status epilepticus, which is really rare in the following puberty. Because yes, I mean, following up our patients who became older and reached puberty, probably there is an improvement during puberty that might really go well with your theory, Joseph, on reaching puberty and changing the hormonal status. What is really puzzling instead is the MRI findings, which are completely negative. I mean, there's really nothing on the MRI that can be observed, despite the possible function of the PCDH19. These patients might probably develop some cerebral atrophy, but then again, they have quite a few seizures, so that might also be related to treatment or to seizure frequency. So it's quite puzzling. Another thing, I think the connection with the vaccination and so on, it's just related to the age of seizure development.
That's
There's really nothing to do with the development of PCDH19. It's just the age of the coincidence of the vaccination with the onset of seizures.
Yeah, certainly it can be coincidence, but I just mentioned it in the context of the stressor, which, of course, any vaccination is a stress into an organism, which may have...
With Dravet syndrome, it's the same thing. I mean, it's just a stressful situation, but it's really not related to the vaccination, yeah.
Not per se.
Yeah.
That's correct. So sorry if that information came across that way.
Joseph, I would like to know if it is possible to dose the neurosteroid in peripheral blood.
Yes.
Do you think that we...
That's what was the one figure on.
Yeah, and do you think that it will be interesting to have a program of a systematic dosage of this neurosteroid in affected females or not?
Well, I would love to do that. That's why we sort of introduced the technology. The only problem is that in Australia we don't have as many cases. So I think there are our colleagues here in Rome who are probably much better positions to do it, and I believe they are to some extent doing it. So maybe Nicola can comment on it very quickly. So we would love to see that confirmed. We would like to that validated, and perhaps make some correlates whether that would relate to the severity or how it's going to change even during the clusters or not, or before. So, but yeah, it has not been that easy, at least for the time being. But, maybe Nicola can comment on that.
Yes, thank you. Just wanted to comment about the blood examination of the neurosteroids. We are starting now to measure the neurosteroid in the blood of patients, and also with a control group, and we will perform several measurements within the day, following the following stimulation induced by ACTH. So we will see data.
So I guess I don't completely understand how you see protocoherine 19 linked to the steroid synthetic system, and why there isn't a problem for boys. And I think I missed something in the talk, so maybe if you can take me through that again.
Yeah, I'll try to do my best. This is really on mind of all of us and everybody else, but drawing parallels perhaps with the CFNS. So these cells do contact each other. PCDH19 is a cell adhesion molecule, but we believe have other function. Probably it's a signaling molecule as well. So these cells do contact, that's our hypothesis, each other, but these contacts become less stable, and as a consequence there's different sort of gene expression downstream of the protocoherine. That's exactly happens in the CFNS and in a mouse model, that these cells with different Ephrin B1 expression, either present or not, they do contact, but the contacts are less stable, and as a consequence these cells migrate differently and become different, and you can see it as that's the result. actually of the disorder. Now, of course, why the males are not affected, I believe that there is a compensatory mechanism. We have seen it in eukaryotic cells in many times. We have multiple genes, the whole genome went through 2 rounds of probably duplication. So there we have other protocoderins, 17, 18, which are really right in the same group. Actually, protocoderins 17, 19, and another protocoderin are the ancient duplicons. They have the diaphanous gene right next to them. So this is the same locus which contains this protocoderin, which has been triplicated in the human genome. So my hypothesis is that the boys or the girls which don't have either copy of the PCDH19 would be simply saved by other protocoderin to large extent, or to 100%, because there is alternative answer, and I think that's why the eukaryotic cell, we as an organism survive, because we have alternative answers to these difficult questions. Now, why is such a still conserved protein if boys survive? I think the selection is at the level of the women. That's where probably the selection is very important, which maintains the highly conserved nature of this protocoderin. I don't know whether that explains the question, but it's my perhaps understanding what's going on.